Okay, I don't put on the microphone. I just want to say that I'm really grateful and it's a real privilege to be here. That I was invited, our lab was invited to present here. And what I'm going to talk about is a lot of people's favorite class of antifungal drugs. But I'm going to talk about where they came from. And I'm going to talk about how fungi put these molecules together, and maybe even at the end show some data might even suggest what their function is in fungi. We know what their function is in the clinic, but their actual function in the fungi. So I'm going to go ahead and get started by showing the natural product starting point for caspofungin acetate, and it's a molecule called Echinocandin B. It was discovered at Merck. We all know, how great it is, and we all know how much money it makes for Merck. And so this is the crude natural product, and then in a 3-step manufacturing process, of course in the lab developing it was much more complex. They take off this hydrogen from the ornithine, put this diamine on here, and they get rid of this keto group, and this gives you, and this complex with acetate, and this gives you caspofungin acetate. All right, so I'm going to talk about how this molecule is made. And this is, wait a second, there we go. All right, so not too long ago, our lab published the biosynthetic gene cluster for pneumocandins. And it's a huge set of genes. It spans about 100 kb. And the core anchoring gene right here is a non-ribosomal peptide synthase. This makes the core peptide. There is a polyketide synthase that sits adjacent to that makes the dimethyl myristate side chain. And then there's a bunch of oxygenases, P450s up and down here. There's a transporter. And then the pneumocandin pathway, the echinocandin pathway, is really 2 pathways in one. It also has an embedded pathway for the biosynthesis of homotyrosine, which is really a very strange amino acid. And this is the only place that I know of in a fungi that this amino acid occurs. It does occur in cyanobacteria. Okay, so we were writing up our paper to publish in BMC Genomics, and we were going to start writing the paper. And Yitong's lab at UCLA published this paper in JACS on the echinocandin B pathway, and it's remarkably similar to the pneumocandin pathway, but it does have some important differences. So here's the core anchoring gene here, the non-ribosomal peptide synthase that makes the core. A bunch of oxygenases, P450s up and down. It has the same ABC transporter. And what's strange is there's no PKS. They believe that the side chain, the linoleic acid side chain, comes from the cytosolic fatty acids. And then on a different scaffold in their genome, they found the homotyrosine biosynthetic gene cluster. Okay, so this is very similar, but it's different. Okay, so let's go ahead. So I'm going to show you how we think pneumocandins are put together. This is, god-awful biosynthetic pathway, but it's really quite simple if you look at it closely. So you have a polyketide synthase that hooks acetates together. Some methyls are put onto here by a methyl transferase. And then there's a ligase that shuttles this polyketide to the first stylation domain of the non-ribosomal peptide synthase. And then ornithine's added. This is really important to pay attention to. There's a proline, gives you hydroxyproline, that goes in here. Then the homotyrosine pathway feeds in here, gives you homotyrosine. Wait a second, okay. Then there's glutamine, and there's an enzyme involved in hydroxylating the glutamine. And the second proline does not come from proline. It comes from leucine, and there's some oxidation reactions that cyclicize the leucine to give you this protein. So these have different biosynthetic origins, and this is really important. And I'll explain here in a little bit why. And then there's a condensation domain that closes this up. And then there's a couple more P450s that come in here, oxidize this ornithine, and then oxidize, put an extra hydroxyl group here on the homotyrosine to complete the process. Okay. So we've done most of the knockouts of most of the genes in the pathway to understand how this works, and I'm just going to show you a couple examples of this. The main tool we use to do this work is we use agrobacterium-mediated transformation, and we basically insert a hygromycin resistance gene right into the target gene and then watch chemically what happens. And so I'd just like you to look at this HPLC profile here. In wild-type Gloriollothiensis, the ascomycete that makes this compound, the dominant molecule is pneumocandin A0. Okay, and the minor is pneumocandin B0. So this was a big problem at Merck because the B0 is more potent. It was the one they were interested in taking forward as the candidate. So they had to, not only to get the titer up, but also to simplify the purification process in the pilot plant. So this was so to get this ratio changed, they did classical mutagenesis work and medium optimization work to get this to change the ratios. So there's this oxygenase that's involved in this cyclization of isoleucine, and so if we knock that out, this is what happens. We just get exclusively pneumocandin B0. Just one simple gene inactivation would have, saved 2 or 3 years of work at Merck. And not only does it give you exclusively B0, but it jumps the titer about 9-fold. All right, so that's really interesting, and I think it's potentially useful, although it's probably way too late for Merck. Okay, so next slide. So just to show you how this works, in the wild type, normally what happens is you get the methylated form of proline from leucine here, and that gets incorporated into the 5th position. And this is the preferred amino acid, but this adenylation site here is sloppy, and it'll also pick up this one too. So you get both products formed. In echinocandin, this is really interesting. Echinocandin, this doesn't happen. You just get this form. So there's something different about this adenylation site here from the echinocandin NRPS. But anyway, so if you just block this oxidation of leucine, you only have the NRPS has this one choice to pick up the proline, and you get exclusively pneumocandin B0. All right, so that's really interesting thing you can do with the pathway. All right, so now I'm going to talk about just a couple more examples. The other interesting things you can do with this information. So there's 2 P450s here and another oxygenase, which I'll call GL-oxy1, and we're just going to knock these out one by one and see what happens, just to find out how this thing works. So we knock out P451, and we get a shift in our HPLC profiles, and by mass spectrometry, we see we lose a hydroxyl group here, and if we knock out P452, we lose 2 hydroxyl groups. Okay, that's really interesting. And so what happens is the P452, what we believe is happening is iteratively hydroxylates the ornithine, all right? And these products are called pneumocandin B2 and A2. These were seen in pilot plant fermentations at Merck. So there is some error in the actual natural fermentation that causes this to happen. P451, you get the C4 hydroxyl group missing, all right? And then you get the pneumocandin A version of this and the B version of this. So you get two analogs here. And these are new. These have never been seen before, all right? So, okay, so now we're going to knock out GL-oxy1 and to see what happens. And this was pretty crazy when we did this. It was the simple gene inactivation, and we get all these peaks. A and B's disappeared, and then we get all this stuff. So Yan Li, he methodically and persistently isolated every single one of these, and did the mass spectrometry, and you see a whole range of molecular weights here, from losing one hydroxyl group to this one here where you lose all four. Okay, and you get every permutation you can imagine. And so this gives you lots of different permutations of hydroxylation to look at the structural activity relationship here. Okay, so why does this happen? Why do you get all these different variations from one single end of activation? So what we think happens here is if you take off this hydroxyl group here, this is a fairly flat structure, and this somehow changes the conformation of the ring, and you get this erratic hydroxylation from the other P450s and oxygenases. So it's hit and miss, and you get all these different variations produced. Okay, so you get at least 9 different permutations here formed at one time. But it gives you a lot of material now that you can look at the effect of hydroxylation on the actual bioactivity. So we did that, and so Jan Lee accumulated all these compounds, got enough to do the test. And we're a small lab, we don't have the capacity to test lots and lots of isolates and lots of strains, but we took representative of some of the major pathogens and just titrated these down. And so pneumocandin A and B0, there's B0, here's A0, so that's our reference here, amphotericin here. And then these two here that we're going to call I and H, which are the C4 deshydroxy forms, are actually more potent than pneumocandin B0. So potentially that's useful. And there's some precedence for this because there were some patents from one of the German or Swiss pharmaceutical companies, I think it was Herx, on Echinocandin back in the early 2000s. And they found a form of the Echinocandin that was missing this hydroxyl group, and they claimed it was more potent than the normal echinocandins. So there was some precedence in the literature for this observation. So this is potentially useful and interesting. Okay. Okay, so let's talk about the side chain a little bit, the various side chain diversity in the family. And the side chains are important because they really impacted, in one way, the development projects at the 3 different pharmaceutical companies that developed these drugs. And the reason this was important is because these straight chain fatty acid type side chains that were apparently come from cytosolic fatty acids were very hemolytic, all right? And that necessitated medicinal chemistry to find a replacement side chain. There's also a side chain called Echinocandin, and it has a methyl group on it. And you would think this might come from polyketide synthase, but actually it doesn't. It probably there's no polyketide synthase in this pathway. So we think it's this fatty acid synthase here is using some kind of branch starter unit. So we need to figure that out too. That's another project down the road. And then the Merck molecule that started with the 10,12-dimethyl myristic acid, this is much less hemolytic than these are, and less hemolytic than the MIC of the pneumocandin. So it really wasn't that big of an issue. So they left the native side chain on the molecule. Okay. So here's what the replaced. You all certainly know these. This is palmitic acid was cleaved off here, and they put the synthetic side chain on here to generate mycofungin at Stellus. And the same with Echinocandin B. The side chain was replaced with this synthetic side chain here to produce indigula fungin. All right. The other interesting variation in this family that impacted the development was the discovery of the sulfated echinocandins. And these were discovered at Stellus. It used to be called, this is what they were called Fujisawa, I think, at the time. And we observed these compounds also at Merck. If you look in the right place, you will find them fairly commonly. And what's important here is not only did it, because the sulfate was novel at the time, and it opened up patent space for them, of course, but this increases water solubility by about 10-fold. And pneumocandins and echinocandin B are not especially soluble, So this made their starting material was much more soluble and gave them a really good starting point for their program. All right. The other thing I want to point out, these are only made by some of the echinocandin producing fungi. And they're in this group of fungi we call the Leotiomycetes. They're restricted, so far as we know, they're only found there. And the other thing that's interesting about this is sulfated natural products are rare to begin with. And fungi, they're really rare. I can think of some sulfated terpenes. But this is the only example that I know of a non-ribosomal peptide that's sulfated. Okay, at least in the fungi. And so there's some questions about what's really happening here. And so what we hypothesize probably is you have the tyrosine hydroxyl group here. Somehow there's a hydroxylation group here, and the sulfate gets esterified to that hydroxyl group. And sulfation is probably moderated by some kind of aryl sulfotransferase. What we don't know is whether it sulfates the tyrosine first and then homotyrosine version is synthesized, or it waits until the homotyrosine is synthesized and it sulfates directly the finished product. That, with the sequence, we don't know yet, so it would be interesting to figure that out. We do know that when this is synthesized by the cultures, apparently the reaction goes to completion, because you don't find residues of pneumocandin, unsulfated pneumocandin in the broths. So it seems whatever is happening, it seems to go to completion. So that's interesting too. All right. So we sequenced one of these fungi, the one that produces this. This is an interesting molecule. I'm just going to call it 293 for this, to be brief about it. And basically, it's pneumocandin with a sulfate on it. All right, it has the same side chain, same amino acid sequence, and it has this sulfate on here. So we sequenced the genome, and we got the pathway out, right? And it looks pretty much, there's some genes that are rearranged, but it looks pretty much like the pneumocandin pathway. There's no obvious candidate for a sulfotransferase here. However, we started doing some blast searches and some modeling, and we did find a putative aryl sulfotransferase on one of the other scaffolds. And we went to model this, and surprisingly, we thought maybe we could use a human sulfotransferase as a model to figure out what this might look like. And it has almost no homology to the human sulfotransferase. But it's quite similar to some bacterial aryl sulfotransferase, and we used an E. coli aryl sulfotransferase to try to generate a model. So we also think there's a hydroxylation involved in this, so we got to figure out where that gene is also. So there's an oxygenase upstream of this aryl sulfotransferase, and there's the P450 here. So these are likely candidates for the hydroxylation part of the reaction. So right now this experiment is in progress, and we're waiting for the transformants to come up on the plate. And so in a few weeks, hopefully, we'll know if knocking this out will desulfate this molecule. Okay, so the experiment's ongoing right now. Okay, so another really interesting aspect. I was trained as a mycologist, and I think like a mycologist, and so for me, one of the most striking things about the pneumocandins and the echinocandins is you only find them two places in the fungi, and they have a really peculiar distribution. Unlike a lot of other natural products from fungi, they are somewhat widespread distributions, or radically spread across the whole fungal kingdom. These have very, very narrow focused distribution. And you find them in certain aspergilli, and then you find them in a whole group of really obscure Leotiomycetes, including our Merck's Gloria lothiensis, and the Astellas mycofungin comes from a fungus in the genus Coleophoma. And there's some other strange genera in here too that make these things. And I believe this is true because people have been looking for echinocandin-like molecules for 40 years. and been looking in fungi. And this is where we find them, always. They always come back to these two places, one of these two places. And the other thing that's strange about this is I don't know any other natural product family that has this kind of distribution. They have other peculiar vicariant distributions, but this is absolutely unique. Okay? And the other thing about this is I'm just going to point out this date. So these are the Leotiomycetes here and the Eurotiomycetes here. These two groups separated some 300 some million years ago. All right, so how did pathways that look that similar get in these two groups of fungi, distributed between these two groups of fungi? Okay, so let's go back and look at structural diversity again. We're going to ask the simplest, if you want to just simplest type of echinocandin might be a culeasin. Palmitic acid side chain, very simple. Relative to something like 293, which is sulfated pneumocandin. Polyketide side chain, sulfated. This, to me, is the most complex. Okay, so we've sequenced genomes of most of the representatives of these echinocandin producing fungi. There's been a few, like the Aspergillus aculeatus, the genome was at JGI, so that saved us some money. So if you start to look at the orthologs and the pathways, you see the common shared genes in this, among these pathways. You start to see the chemical logic of how these different variations are encoded by the different genes. And the other thing that's quite clear here is there's a large hiatus in homology between the ones that are in the Leotiomycetes and the ones that are in the Aspergilli. Okay. And you can actually line up these gene clusters, and now I have a classification system for echinocandins, echinocandin family. So the big first dichotomy is between the Aspergilli and the Leotiomycetes. So here we have the number 5 positions, either serine or threonine, and here we have glutamine, which is then hydroxylated. So this is the main hiatus. And then you get the sulfated versions, and then you can see things in like this, that this particular gene cluster lacks a P450, and this is the one that hydroxylates that C4 position on the homotyrosine. So you can classify these things now. Okay, but the important thing is this hiatus here. And so there's so this is, these kind of things make you question where do these things come from. And one of the ways we examined this was we made a phylogenetic tree from the adenylation domains of the non-ribosomal peptide synthetases. And to do this, we just went back and took some data from this wonderful paper by Karen Bushley and Gillian Turgeon when they made a phylogenetic analysis of non-ribosomal peptide synthetases in the fungi. We just took their data and then added ours, basically. So we root the tree here with the beta-lactam synthases here, and this is americelamide. This molecule is another non-ribosomal peptide synthase that the ligase, I think, is somewhat related to these, but the non-ribosomal peptide part of the molecule is totally unrelated. But the pneumocandin and adenylation domains are monophyletic, beautifully monophyletic. And then what's even more striking is when you look at the tree, the amino acid, the tree is based on amino acid selectivity. It doesn't go by species or phylogeny of the fungi. It's all the threonine or serine selecting A domains are clustered together. All the proline selecting A domains are together, on and on down the line. So these things all had a common evolutionary origin. And then there's one thing in here that is not an echinocandin, and it's this non-ribosomal peptide synthase called ENPB. Nobody knows what that is, the product of that is. It's an Aspergillus nigrans, and it's a bimodular non-ribosomal peptide synthase. And so we don't know what the product of that is. But that could be perhaps the origin of this non-ribosomal peptide through synthase through domain replication. So if we go back and do this exercise for every single gene in the gene cluster, we get the same result. So just to give you an example here, we took all the ABC transporter genes we could find, and we made a giant phylogenetic tree, and then put in the ones from the echinocandins, and the echinocandin ABC transporters are monophyletic. The acyl-CoA ligase is monophyletic among all fungal acyl-CoA ligases. And it goes on and on and on down. The same with the enzymes in the homotyrosine pathway, the oxygenases, and the P450. So these things all came from the same evolutionary origin. So what we think, this is pure speculation now. There was some ancestor, back 300 some million years ago, and by magic, we got a homotyrosine pathway come together with an acyl-CoA ligase and some type of non-ribosomal peptide synthase. All right? And supposedly included the oxygenases and the P450s also. And it probably went into one of these lineages very early on. It either went into the Aspergilli, and you can argue it was the Aspergilli because some of the closest relatives were in the Aspergilli, and this went from Aspergilli into the Leiotiomyces by horizontal gene transfer. And then there's these additional functionalities recruited, and they're PKS. And then coincidentally, it got stuck into a genome that had this really nice active aryl sulfotransferase. But you could argue the reverse, that this moved from here, this started out complex and then moved to the Aspergilli, and it lost functionality. And it was also this mutation at the 5 position that switched from glutamine to serine or serine to glutamine, whichever way you think it went. So that's what we're thinking right now. Okay, so that's the end of that part of the story, and I just want to finish with this one really odd observation, and this is really a great audience to show this. This is very, very new data, and we don't know what it means, but I'm going to throw it out here and show it to you all, and then hopefully you can come up later and tell me what you think it means. So we also sequenced the genome of the fungus that makes this molecule. It's called sporophungin. It was discovered at Sandoz in 1982, or the patent came out in 1982. So Sandoz was working on endophytes and getting molecules out in 1982. So endophytes are not a new phenomenon. This is 30-year-old work, okay? So here's the fungus. Here's the Merck fungus. So you can see these molecules were, almost the same. So Sandoz essentially had pneumocandin before Merck did. And so the gene clusters, here's the pneumocandin gene cluster again, and up here is the sporophungin gene cluster. The microsynteny is the same. And if you go down through here, I don't know if you can see this very, I should have colored it different. Right upstream of this, the P450s, there is an extra copy of FKS1. We saw this, it just blew us away. We couldn't believe it, because there's another genomic copy of FKS1. So there's 2 FKS1s in here, and there's one embedded in the pathway. So we don't know what this means, right? And right now we got to go in and study this and try to understand what it means. But if this is really real, that it's embedded in the pathway, then it indicates to me that these molecules really were evolved to interact with glucan synthase. I was always skeptical, at first, I thought it might be some kind of chemical accident, but this has convinced me that the real function is to interact with FKS1. Okay, so I'm going to finish right there. Just thank you all for coming out late on Friday. This is my wonderful lab here with all the guys in my lab, Jan Lee, our chemist, and Li Chen and Shun Wei have done all the done the genomics and the gene inactivation studies. And Dr. New has also helped us with some of the structure determinations. She's gone back to China now. And I also just want to thank and acknowledge all the people that have helped me learn about natural product screening in general, but also the pneumocandins, including a lot of the people that were in the original pneumocandin development project at Merck. My old colleagues at Merck Sharp and Dome de España, and those that went on to start Fundación Medina in Granada. And then this guy here, Ji Kwang-on, he really got this project started. Okay, here's my postdocs. And then this fella here, Xinjiang Liu, he's been bankrolling the genomic sequencing at the Chinese Academy of Sciences. And then Akihiko Fuji, he's given me a lot of historical information on the sulfated echinocandin, so I thank him also. So I'd be happy to try to answer any questions or take any of your comments, and once again, I'd really like to hear people's ideas on what the second FKS1 gene in the gene cluster what that means. Okay?